organizations that exist to advance innovations, ideas, and processes that make a difference in global health. TIPS extends to over 30 countries and is the key strategic partner for many notable organizations, initiatives, and events. In the past few years, TIPS has contributed millions in scientific impact. You'll find their successes published on their open access journal at www.infectioncontrol.tips. We're also pleased to have the Healthcare Infection Transmission System, or HITS Consortium, as a strategic partner for this webinar series. I hope to see all of you at the next HITS conference. HITS 2020 conference will be held August 4th through 7th at the Long Island Marriott. Register today at hitsconsortium.org. HITS takes a holistic perspective to targeting HAIs. By including multiple disciplines in the conversation, HITS removes silos and encourages a systems approach aligning with infection prevention. This webinar series is sponsored by Crecia, maker of NADCC tablets. Thank you for your support. We're pleased to have Dan Mueller with us today. I'll turn it over to Dan.
Oh, hi. This is Dan Mueller with Crecia. We're really excited to sponsor the webinar series. We make tablets that release HSCL, and we're approved as a sporicidal. And thanks, everyone, for joining.
Thank you, Dan. If you have any questions, please type them in the webinar section labeled questions. With over 500 participants across this 3-webinar series, we will do our best to get to all your questions. We will follow up after the webinar to provide answers for any questions we are not able to answer today. Today's presentation also includes polling questions, and the results are anonymous. Without further delay, it is my pleasure to introduce Dr. Christine Greene. Dr. Greene has over 10 years in the study of environmental survival and transmission of pathogens. She holds a Doctor of Philosophy in Environmental Health Sciences and a Master's in Public Health in Hospital and Molecular Epidemiology. She co-founded the HITS Consortium to bring awareness to the complex system of transmissions across the facility. She's a TIPS board member, and she's convener for the development of international standards for hand hygiene in healthcare. I'll turn it over to Dr. Greene.
Thank you very much, Katie. Thank you. And happy Wednesday to everyone. I'd also like to thank our sponsor, Crecia, and the TIPS organization for providing today's webinar and inviting me here today. And a big thank you to all of you for logging in and for spending this hour with me. So disclosures, I am a regulatory microbiologist. I work for TSG. I also am a co-owner for EVS Protects. I am president of the HITS Consortium and a board member for the TIPS organization. So today we're going to talk about methods for environmental surface monitoring. But before we do that, I think it's really important that we understand what we mean by environmental monitoring and the purpose for doing it in the first place. Environmental monitoring is widely used as a means to be proactive and to prevent the spread of infectious diseases, and primarily in the food and the healthcare industries. It's essentially a set of processes or activities that we're going to do in order to characterize and evaluate the effectiveness of our cleaning and products that we use to clean. Historically, this has been part of a surveillance system for microbiological contamination remediate efforts. Primarily, when we've done microbial sampling in the past, we were specifically looking to recover bacteria and sometimes yeast and mold. But now, over the decades, we've really realized the value of this in terms of preventing or protecting our public health. And as a result, we have a lot of new tools that have emerged as environmental monitoring is expanding from the scope of just looking at microbiologically controlled environments. So why would we want to do microbial monitoring or environmental monitoring? First and foremost, for public health and safety. And over the years, we've had regulatory requirements that have come up as a motivator. So FDA, ISO, the European Union, the pharmacopoeia industry, they all have some sort of a standard or a regulation in place now. And these regulations primarily affect the food and pharma industries for monitoring contamination to protect consumers. When I talk about environmental monitoring, I do want to make note that I'm referring specifically to biological, not non-biological. So I'm specifically referring to infectious agents. Today I will talk about that, and we really want to be careful to understand that there are multiple sources of contamination that come from all sorts of areas in our environment. Contamination comes from cough droplets, hands, dirty surfaces, and the water we drink, dust, and all of those sources can contribute to the contamination that we find on a surface. And also, pathogens can survive on surfaces for very, very long periods of time. And this is part of the reason for this is that they are often resistant to chemicals, and they can also be resistant to UV and other methods of treatment. So we can see in this chart here on the right, I show you the data for some bacteria and viruses, and you can see some of these bacteria, such as Acinetobacter, they can survive for months. We've got Enterococcus that can survive for years in the environment. Now, viruses can also survive in the environment for quite some time. They're a little less hardy, right? But they can still survive outside the human host to some extent. Now, I will make a comment that specifically for viruses, over time, they do lose their infectivity as they are in the environment. So they're most infectious in those first 24 hours. And then that infectivity does decrease over time. And that varies by the pathogen or the microorganism. So it's really important to know what the definition of clean means, right? What are we evaluating when we're doing environmental monitoring? This is the CDC's definition of clean. They say it removes germs, dirt, and other impurities from the surface or an object. But what's interesting is they specifically state that, one, remove is a word that they repeat here throughout. They specifically state that it involves soap or detergent and water. So wiping across the surface with a dry cloth or plain water does not result in what the CDC would classify as clean. And they also specifically use the word process to physically remove germs from a surface in order to lower their numbers and lower the risk of spreading infection. So the point I want to make here is that the word clean does not equate disinfected. So let's talk about that. In environmental microbiology, we define clean in terms of microbial removal or efficacy. Now the graph here shows or compares three of the most common terms that we use to describe levels of efficacy, with the most efficacious level of microbial removal being sterilization. Sterilization destroys all forms of life. It's usually accomplished with very high levels of heat, pressure, such as an autoclave. Disinfection, both sterilization and disinfection are considered to achieve at least 6 log reduction in bioburden or better. The biggest difference between disinfection and sterilization is that disinfection is not considered to kill spores. And it also has a lower level of efficacy in general, such that it eliminates many or all microbial life. Sterilization flat out destroys everything. Disinfection eliminates many or all, and it's not considered to kill spores. So if you are concerned about spores in your environment, certainly you'll want a sporicidal. And there's regulations around that. Then we have sanitization. So this is defined as lowering the amount of germs on a surface, and typically is defined as achieving a 5 log reduction. Sometimes it is as low as 3, depending on industry or the regulation, but sanitizing has a much lower level of efficacy. So the point here is that while the term clean is not specific to microbial bioburden, these words, sterilize, disinfection, and sanitization are. All 3 of these words are by the amount of germs or microbial life that are killed. So when we think about environmental monitoring, you have to know what you're monitoring for. Are you monitoring for cleaning, or are you monitoring for disinfection? We don't have an official standard in this sense, but we do have some tools at our disposal. So we're going to talk about how we can measure our whichever definition of clean that you're trying to achieve. All right, so we have about 3 basic levels of cleanliness that we can assess. We can look at the appearance of clean. Does it look clean? We can ask, did my staff actually get the work done? Did they clean the surfaces? And then we can ask, was the surface disinfected? Did we achieve our intended log reduction goals? These 3 questions, they'll span the spectrum in terms of our ability to assess risk of disease transmission. So for high-risk areas, such as an operating room, we might want to ensure disinfection. But in a waiting area or somewhere where the risk of disease transmission is less of a concern, you don't have high-risk population in that area, for example, then perhaps ensuring that the area was cleaned is all that's necessary, right? So for each of these levels, we have a monitoring tool available. The key is to know when and how to use which tool. If we want to ensure that the appearance of clean is maintained, and believe me, I know in the U.S. this is an issue for many of our hospitals with respect to the reimbursement system, then visually cleaning, you just need to do a good visual inspection, that should suffice. If we want to confirm that the staff actually cleaned the room, then we can incorporate things such as fluorescent markers or bioluminescent sampling. And then if we want to, the next level is if we want to confirm that the surfaces have been disinfected, then we're going to want to use microbiological methods. So as I talk to you today, I'm going to focus on ATP, microbial testing, ATP testing and microbial testing. Both of these areas or methods, I find a lot of confusion and misconceptions. Both methods have strengths and they both have weaknesses. So it isn't so much a matter of which one to use, it's more a matter of when to use which one. So first, let's talk about ATP. So ATP bioluminescence monitoring, this is really, really popular. Most of you on the line here are probably very familiar with it or use it because this provides very rapid on-the-spot results. In a matter of less than 5 minutes, you can take your sample, perform a fairly simple squeeze and shake procedure, slip it into a handheld reader, and then boom, you have your result. It's a great tool. It's especially great for training. It's great for monitoring whether or not a surface has been cleaned or not. And I'm going to explain to you why. And then we're also going to talk about how this test works. But most importantly, what I really hope is that you understand and learn what that RLU reading that you get on your meter, what that actually means. What does that mean to you? So first, we have to give a little bit of background information just to make sure we're all up to speed on the same page. So ATP is a molecule called adenosine triphosphate. It is made up of 3 phosphate molecules, a sugar molecule, and then a genetic base, adenine. This molecule is manufactured by all living cells, and it is, I'm sure you've heard it called the energy currency of the cell. This is what gives our cells the energy it needs to replicate and do all its fancy things. ATP meters, they provide you this real-time quantitative measurement of the presence of this particular molecule on a given surface. The theory is that I have ATP present, well, then there must be cells or of some sort left behind on the surface, meaning that the cell or the surface is dirty. Because, you know, we find ATP in bacteria, chloroplasts, so plants, and then in all human and mammalian life. How does it work? So the ATP system has 2 really important chemicals in it. It has a releasing agent and an enzyme. So the ATP releasing agent, if you look at this graph here, it interacts with the cells that you pick up on your swab, and it acts like a detergent by breaking that cell open, which is needed in order to get the ATP out of the cell. So now you have ATP floating around, and ATP and luciferase. Luciferase, this technology comes from the bioluminescence that happens with fireflies. The ATP and the enzyme, they react. They go down this nice little enzymatic pathway to produce a form of light, okay, or bioluminescence. And that light can be measured using a lunometer that is in your little handheld device. And the concept is that the light that you get in your reading, or those RLUs, is directly proportional to the amount of ATP present in your sample. So I have a polling question. Dan, you want to put the polling question up? All right. The question is, ATP monitoring systems measure the amount of cells on a surface. Is this true or false? All right. ATP monitoring systems measure the amount of cells on a surface. The answer is false. So those of you, 61%, good job. And if you answered true, that's okay, because that's one of the reasons why we're here today. All right, so RLUs are a unit of measurement for light or bioluminescence, not cells, okay? RLUs do not equal cells, and let me explain why. First, the bioluminescence comes from a reaction that involves ATP, which is a cell constituent. So yes, there is a correlation, but it is not a measurement of cells directly, and certainly not of bacterial cells. And you'll learn why. So the concept is that there's a direct correlation, and it shows in this right-hand graph here. The concept is there's a direct correlation between ATP and RLUs. This is correct. The leap that people try to make is that this means that there's a direct correlation between RLUs and cells. That's not correct. In the lab, which is a controlled environment, you might see this, but not in the real world, because the amount of ATP that is contained in a cell depends on the type of cell that it is and its metabolic state. So we have to remember that ATP is an energy molecule of the cell. So a dormant cell that is not producing very much ATP, or another otherwise inactive cell that's not growing, such as perhaps a spore, would have very less ATP compared to an actively growing cell. Also, dead and dying cells, such as those that have just been exposed to UVC exposure, these cells will, as they die, release ATP. We call this extracellular ATP. So for example, UV exposure breaks those cells open, they're on the surface, they die, they're broken open, the extracellular ATP is released. This is also picked up and read by that meter. So the ATP test does not differentiate between live cells or dead cells. So if you're looking to see if the surface was cleaned, it may not be too much of a problem. But if you are trying to measure cleaning efficacy or validate disinfection, then this is not the test for you. And I really hope that that made sense. So just to help give you something to chew on, a little bit of evidence that I have. I did a correlation study a couple of years ago when I was at NSF International. And the purpose of this, first it was a field study, and the purpose was to evaluate a new product that I had discovered. And on the market at that time, this product purported to be a tool to evaluate disinfection or contamination levels on surfaces in a very rapid way. The way it works is that you take your sample like you would any other ATP sample, but you then incubate it using a little desktop incubator for about 6 to 8 hours. Then you take it right out of the incubator, you use the ATP technology, and it would give you an RLU reading that should correlate with the amount of bacterial contamination that was on that surface. I was really excited about this because I thought it provided just a wonderful option that was much more rapid than standard methods. What we did was we compared this bacterial ATP test to the same manufacturer's provided standard ATP test. And then we compared that to microbial swab samples. You should know that all of the samples that we collected were from a large university hospital, and we compared 394 standard ATP samples to 394 bacterial ATP samples to 394 microbial samples for a total of 1,182 samples collected. And we collected these samples in the early hours of the morning before their cleaning staff got in and did any cleaning. So the results that you're looking at here were from uncleaned patient rooms. And we found no correlation between ATP and colony forming units in general. There's no correlation. And what was interesting to me was that the standard typical ATP system did a better job than the quote-unquote bacterial ATP meter. So, you know, you can see the blue line on the left, that's the specificity line. It shows how specific the product was for recognizing contamination. The bacterial ATP meter was pretty bad when there actually was contamination on the surface. It actually, the reading was, the cutoff threshold for that particular meter was a RLU of 2. And 85% of the time, the RLU read less than 2 when in fact there was contamination present. Now, the ATP system did do better. 50% of the time, the sample was identified as clean when there was actually contamination on the surface. That's still not great, considering these really were dirty rooms. We knew that it had been 7 or 8 hours since they'd been cleaned. So it just gives you evidence and just, you know, helps to support the case to encourage you to be really careful with using these ATP technologies for assessing disinfection. Especially as newer, more novel products come out on the market, you just need to do a little bit of homework. So our next polling question. There we go. ATP monitoring systems measure, nope, that was the old one. We need the next one. Can the products that you use to clean a surface, your cleaning chemistry, so it could be anything, can that cleaning chemistry impact the results of your ATP reading? So if you think, you know, it kind of does, but not very much, I mean, you just put it in through a bucket of either yes it does or no it doesn't. We'll give you about 10 minutes, or 10 minutes, we'll give you about 10 seconds to answer that. Okay. Oh, I'm so happy to see that you all said yes. The majority of you, yes. Okay. So you guys are so good. All right, let's talk about that. Yes, yes, yes. The answer is yes. Depending on the cleaning chemistry, the disinfectant residues that are left behind can definitely impact your measurement. And honestly, that could have been a factor in the study results that I just showed you, because we didn't account for that. This was just, that study I just showed you was just walked into a facility, found out what type of ATP meter that they're currently using. Hey, great, it matches up with this one that we want to test, and we tested it based on, what they do every single day. So that could have impacted those results, right? Anyway, from my experience in healthcare, there's not a rinse step after the surface has been cleaned, right? And that's standard practice for the food industry. So let's talk about why we have this interaction. All right, this is a study that looked at the effect of disinfectant residues on ATP results. And this is a Canadian study. It's published in PLOS One, 2014. What they did was they took these 4 ATP manufacturers, Kikkoman, 3M, Hygiena, and Charm, and they tested it, each one of them, against 14 different disinfectant products listed here on the left. And I provide you the DOI if you want to look this up. Anyway, these data that you're seeing here, okay, so they looked at the deviation of the average ATP reading from a control sample. Now, the majority of the results were quenching, so they made those a positive number. So the negative numbers are the enhancing effect, okay? Now, I caution against using this data to go back home, back to work and say, I use the 3M product in Cabicide, so I need to reduce all my results, so my RLUs by 83.67%. That's not what this is for, and that's not what this is telling you. These numbers, the purpose of the study was to look at the interaction. Does the interaction exist? And to prove that your disinfectant can impact either in a positive or in a negative way your ATP results. So yes, we need to do more work so that we can make this more translatable and find more generalizable figures for us, but that literally does not exist right now to this day. So, but the point of my showing you this is that yes, this is a real thing and it really happens. And the best way to understand it is to go back to that basic chemical reaction that's taking place and the 2 primary components in the ATP test, your enzyme luciferase and the ATP releasing agent, okay? So as it turns out, the chemical composition of your disinfectant in general is often really similar to that ATP releasing agent, okay? And the releasing agent right here, so these end up being very similar in chemistry. The releasing agent, which is needed to break that cell open, is similar in chemistry. So both of these products combined are now effectively increasing the total concentration of this chemical type in the reaction. That then interferes with the ability for this reaction, or for this enzyme, luciferase, to actually work properly, right? So you have this overall concentration of releasing agent in the reaction now. It's been magnified by the use of, by the presence of your disinfectant residual on your swab, interferes and adversely affects the enzymatic pathway for fluorescence generation, and then you get those oddball numbers in your final reading. So definitely can lead to a situation where the effectiveness of a disinfectant or your cleaning process is just interpreted improperly altogether. And I hope that made sense. There's a couple of other more points that I wanted to make. There's a lot of variability in the sensitivity of one manufacturer system to the next, okay? And the more variability you have in your results in general means is simply it's less consistent and therefore less reliable. There are differences in reliability between systems, and also differences in their ability to detect gram-negative. There's differences in the software, which then can lead to differences in their detection limits. It's interesting to know that back in the day when ATP technology first came out, and this would be, I believe it was somewhere in the 70s, that these tests were actually done in the lab with standard controls. Then they moved it out of the lab so that you could do this by yourself, on the spot. So there's no standard test or control that is developed at the same time that you're doing your testing. And so you have no true results to compare your results to. So all this, the convenience of on-demand testing and instant results, this all comes at a cost of reduced reliability. And each ATP testing device or manufacturer brand, they read on a brand specific, and really it is an arbitrary scale of ROUs that really results in a overall wide dissimilarity between manufacturers in terms of their threshold levels. But now all of this information, which sounds like doom and gloom, doesn't mean the end of ATP. There is definitely something that you can do to ensure good ATP data. First is understanding what that ROU represents, what it means, and how you can interpret it. Hopefully, I've shed some light on that for you. And then this is what you can do. What you really should do is establish your own ATP threshold for your facility based on the products that you use to clean and disinfect. So these, I personally am a proponent of baseline readings that are established per facility. You definitely probably should not assume pass-fail thresholds are the same from one facility to the next. Usually that's because there are differences in chemistries that are used, sometimes differences in process. But first, I would definitely check with the manufacturer and know what their quality standards are for their ROU thresholds. I've seen them range anywhere from an ROU of 100 to 500 as far as a go no-go criteria. And I gave you this link. This is a study that's published, and they do a really, really good job describing a process. It's an algorithm that you can follow, and it's all collecting samples at certain times during your cleaning process to help you develop what's specific for your facility in terms of a threshold ATP reading. I think that if you can go through this, take the time to follow this, it's sort of intense, but definitely worth it. There is a general assumption out there that if your reading is 2 to 3 times greater than your background ROU values, that then this is a go, no go for what you're testing for. I strongly discourage that. I've never seen, and maybe I'm wrong, but I have personally not seen any data to support this. It is an assumption, and I would be careful against arbitrarily pulling a number out there, especially in a hospital. And if you're in the market and looking to purchase an ATP system, one of the things that I want to see in terms to help me make my decision on which one would be the best to use is their sensitivity and variability data. I think that that's really important, and they probably do provide that in their sales pitch, but if they don't, certainly ask for that. Okay. And I think this is our last polling question. And the question is, ATP systems can detect viruses indirectly, of course, but is this true or false? or only certain manufacturer systems have this technology. So it's either, yes, they all can, no, none of them can, or certain ones can. And by the way, this little image here is our famous coronavirus. All right, so what we see, we have 18% of you said that ATP systems can detect viruses indirectly. 54% of you say nope, they just simply can't do that. And 29% of you said that certain manufacturing systems have the technology to do it. The correct answer is no. So 54% of you really nailed that one. Let me tell you why. But yep, there's Corona. All right, why am I moving? There we go. Let me tell you why. The viruses do not contain, nor do they produce their own ATP molecules. Okay. A virus can be either a DNA virus, or it can be an RNA virus. They hijack the cell, so they get into your body, they hijack your cells, they take over your cells, they take over all the cell machinery, and then they trick it into producing what that virus needs to survive and reproduce. So any virus that randomly lands out in the environment is not producing ATP. It doesn't have any of the components that it needs internally to make ATP. I have had heard of people being up on stage saying, oh yes, it can detect, and I'm telling you that it's flat out not true. It cannot detect viruses. Only microbiological methods can help you assess your risk of microbial transmission. Manufacturers understand this, and they know that they have to have enough of the releasing agent in their system, but not too much, right? And that releasing agent is needed in order to break open gram-negative cells. So they're aware that they have to have this balance. They want enough in there to break open gram-negative. So that's why you hear that some systems are, or systems in general, are limited in their ability to detect gram-negative cells on a surface. There's been a lot of studies that conclude very poor to no correlation between ATP and microbial contamination. There's a study by Dr. Elaine Larson, literally states that RLUs only provide an estimate of gross general levels of cleanliness. So the point is that ATP monitoring is a dirt test. It is not a bug test. So if you need to evaluate efficacy of cleaning processes or the ability for a product or a device to kill microbial bioburden, then you should sample for microbial bioburden. So let's talk about that. Microbiological methods. There are two basic ways that you can sample the environment using microbial methods. Swabbing, using a swab, and contact plates. Both work just fine. I personally use the swabs. The contact plates, and that's shown here, this flat plate. So that's great if it's a flat surface, and those are the only types of surfaces you are looking to monitor. But if so it doesn't work that well. It's a little bit difficult, maybe on a patient bed, on sinks, on door handles, anything like that. The swab just does a much, much better job. I like the ability to get into those grooves and crevices where I want to know that, hey, I want to know that this was disinfected, for example. So yeah, I typically use the swabs. I'm going to focus on that because that's what I have the most knowledge on. So let's move on. So microbial swabbing allows the detection and the quantification, as well as the presence of just a range of, really a wide range of microbial contaminants. This can be quantitative, where we are looking at colony forming units. I'll explain what that means. It can be qualitative. Was it there or was it not there? That's like presence, absence. And then we can do genus species identification. Which always scares people, I think. All right. So what would you sample for? There are three general types of analysis that you would need or could consider. And it's nice that you have these different options. In most cases, total aerobic colony counts right here. This is all that you need. This is not only the least expensive microbial test, but you can typically get those results back in about 2 days. If you're mitigating an outbreak, then you might want to look for specific microorganisms, pathogens such as methicillin-resistant Staph aureus, vancomycin-resistant Enterococcus, C. difficile. All of those you can typically look specifically for, and then there's the identification of those microorganisms that is usually, well, there's whole genome sequencing way too expensive for identification. MALDI-TOF is very rapid and it's an affordable method. This will tell you the genus and the species. So it does not tell you how much, it just tells you in that sample, we found these bugs. So you get a frequency measurement. For total aerobic colony counts and even specific microorganisms, you're going to be able to get how much was there. This here is a plate, and each of these little dots on that plate, that's a colony. And we count them, and they're colony are also called colony forming units. We count those, and we get a number. Also, you can do presence absence. Ooh, went too far. Okay. If you're going to look for a specific microorganism, you're going to want to make sure that the lab that you're sending your samples to can test for that specific microorganism. Most have the capability. So quantitative versus qualitative, I just want to touch base on this because most of the people I talk to, they've never been in a microbiology lab and they really do not understand, what is actually, what are they actually getting in terms of the results? What does it mean to them? Quantitative are colony counts. You'll often hear the word aerobic colony counts, ACC. So it's all general bacteria that's present, that can survive in the open environment. So aerobic is colony count. And it's reported as CFUs, colony forming units per sample or per centimeter squared. So there's machinery that can do this now, or a robot, but this image shows you, they have all these colonies and they're going to count each one, and then you get colony forming units, and that can be converted over to colony forming units per sample and then per centimeter squared. You need that number if you want to calculate log reduction, or if you want to calculate the percent reduction in bioburden. To my knowledge, there's no other way to calculate log reduction, percent reduction in bioburden unless you have CFUs. Now, qualitative simply tells you growth or no growth of bacteria. And there's one particular test that's kind of popular, and it is the banana broth test for C. difficile. And it's a color indicator. And it would look similar to this, but it would be pink if it's positive and yellow if it's negative. So it's a color indicator that tells you it's present or it's not present. In general, they're looking for a word, it's called turbidity. And you can see here in this A, this is an image of basically sterile broth. And then you put the swab in and you swirl it around, and then you look to see, is anything going to grow? And when you get this, you can see the growth. You can see the turbidity of that sample. That's positive, right? And then there's different criteria. So you have more to it a little bit. Like you want to know what's your cutoff for criteria. My cutoff for criteria is if I'm going to tell somebody that their sample is positive, I would want that to be reflective of the level of infectivity. So positive, I would want it to say, yeah, it grew up it's showing us that you probably had quite a bit in there to reflect a high risk. I would, so there's not a fan, honestly, of this side, but it's possible. Report as presence absence. You can report reduction in terms of positive samples. So if you take, I took 100 samples, 10 of them had growth, were present, and you now have a percent. Okay. Benchmarking. So you have your results. Now what are you going to do with it, right? You have to have it to, something to compare it to. There are a few published studies that have come up with maximum CFUs that's acceptable given the risk of an environment. I've seen less than 5 colony forming units per centimeter squared for the food industry. In the hospitals, it is published as less than 2.5 colony forming units or aerobic colony counts. And this is actually quite achievable for the hospitals I've worked with. They actually can achieve much, much less than that overall. And then if you're looking for specific microorganisms such as MRSA, then you definitely want to be less than 1. Important considerations before you collect your sample. Now, I think I only have about a minute left, so I'm just going to go through these really quick. Your swab. You're going to want to pick a swab that's definitely made for environmental surface sampling. I like the flax swab a lot. The flax swab does a pretty good job of picking up bacteria, but polyester swabs, cotton swabs, stay away from them. They are very difficult in terms of picking up bacteria and then getting that bacteria off the swab in the lab so that you can get a pretty good count. Transport media, definitely need to consider that. It's used to preserve your sample in transport, has to contain a buffer to in order to keep whatever is there alive, but yet a broad spectrum neutralizer, because like ATP sampling, you will get disinfectant residuals on these swabs. You want to stop the action of that residual, and that's achieved in your transport media using a broad spectrum neutralizer. There are methods for collecting swab samples. Biggest point here is to make sure that you use a good pressure on that swab and rotate it. Use that head of that swab and the full head of the swab. Make sure you get good coverage of the surface that you're actually sampling. And when you ship it to the lab, make sure you use freezer packs, not dry ice. It'll freeze your sample, no longer good. Don't use ice packs into a Ziploc baggie. It will leak, and your package will end up in a warehouse somewhere, because they're all going to freak out, what's this liquid leaking from this box. And don't forget to include your documentation, and definitely, if you can, ship it overnight to the lab. Get those samples analyzed as soon as possible. Quick polling question, that the variability of soil and bacteria distribution is a concern only for ATP? Is it a concern for microbial testing? Both, or is it just not a concern? And what I mean by that is all this stuff over here that it's not consistently dirty across the surface. Depends on high touch, other factors. The result here is that, yes, I love it. You guys are right on. Both ATP and microbial testing have to deal with this issue. And truthfully, technique matters in sample frequency. So you can address that by increasing your sample size, and certainly do a good job with your sample collection. Now, I knew that I probably wouldn't have time to go through the very basics of developing a surface sampling plan, but you'll have these slides for your own reference, because it's important to have where to collect it, when depends on why, how often you should, and it's all depends on what you need and what to do with your results. But in the end, what you need to think about is, when do you use ATP versus when do you use microbial testing? And this really, really depends on what you are trying to measure. Is it disinfection or is it cleaning? Are you validating efficacy? Is it a high risk or a low risk area? ask yourself those questions. Generally, I think you're going to find that maybe a blend of these two services or two methods is going to help you. My references are at the bottom, and questions.
Well, thank you, Dr. Greene, for this excellent and very informative presentation. We have received several questions, so let's get started. One of the questions is kind of multi-factorial, and it goes back to what you were saying, the slides were available, but how do you establish baseline for pass-fail criteria? Where would a facility start? What guidance would you give somebody that was wanting to start an ATP monitoring type program?
Yeah, good question. So that's on that slide for the ATP. I had a slide there that had, setting a benchmark for your ATP, and there's 4, I think. The second one down gave a link. And that article really lays it out and is very specific on exactly what you would need to do to establish your baseline benchmark for your facility.
Okay, thank you. Can ATP reveal layers of biofilm?
No. Okay, so that's a good question, and it's sort of a difficult question to answer because first off, there's dry biofilm and wet biofilm. Usually, when we're doing an environmental sample, we're sampling dry surfaces in which there may be dry biofilms. I'm thinking out loud here. No, I cannot imagine how it would differentiate layers of a biofilm. And biofilms are composed of a matrix of things. You have your EPS, which is your extracellular polysaccharide substance. There are dead cells in biofilm. There are live cells in a biofilm, and those cells actually, and as you move down deeper into it, you have your dead cells at the bottom and more of your live cells at the top, or is it vice versa? It just depends on where the substrate is for cell survival in there. So I'm not saying that an ATP system wouldn't capture some of it, but I can't imagine how it would differentiate that from other things because it can't differentiate between live and dead cells. And it doesn't necessarily even correlate with microbial bioburden. I feel that I provided a pretty good argument that demonstrates and shows that it's not very good at reflecting microbes in the environment. So I would have to, if it was a yes or a no answer, I'd have to say no.
Okay, and I'll refer that anonymous attendee sent that question in, that all of the master classes will be archived on the HIT website, and we had an entire session on biofilms last week, so hopefully that would be helpful. Our next question is, should ATP testing be performed after cleaning but before UV disinfection?
Yeah, yes. I've had a number of industry people come to me and say, they were having a hard time because people using their product in the field, the ATP results were coming back high, and they're like, no, we know it does its job. How can we address this? And honestly, you address it by not using ATP, so that and that was the answer. So yeah, I would say if you're going to, do it before UV.
Okay. And David asked, how do things like wipes with a 99.9% claim say they are disinfectant when it is only a log 3 reduction? Mm-hmm. And he said it seems like it's more used to clean a surface than disinfect.
Mm-hmm. Right. So that's a really interesting question, and there's a lot of ways that industry gets clever in, kind of skirting around the regulations. So it may have a 99. Wait. So did you say, Katie, it was 99.9, like a 3 log? Yeah.
That's what
it's probably
the question is.
Yeah. It's probably it's registered most likely for the food contact, non-food contact surfaces. So they have to kind of look into, well, what is it registered for? And I guess it's, you know, I just don't know how they can get away with putting disinfectant on the label unless maybe it's registered for another. I mean, you have to look at the full regulation on that, but it should be a sanitizer if it's not 6 logs.
Okay. And going back to your definition slide where you said the CDC definition of clean and disinfectant, so was that for surfaces only or was that for hands as well?
That was for surfaces. I put the link on there where I got the CDC information.
Well, those are all the questions we have today. So I just want to thank everyone again for attending. And if you're interested in receiving CE credit, don't forget to complete the survey by clicking the link in the email you will receive from Michael Diamond shortly. The complete masterclass series will be archived on the TIPS website. And don't forget to register for the HITS 2020 conference. I hope to see you there. And I believe Dan has some closing comments.
Yeah, thank you so much. This is Dan Mueller with Corsia. We're proud to sponsor the webinar series. It will be archived on the TIPS website in the next week or so, each of the webinars, so you can review those. And if anyone's interested, we make NADCC tablets, dissolve into water to release hypochlorous acid, and more hospitals across the U.S. are using our sporicidal house-wide for being proactive for C. diff because it is less corrosive, and we're also listed as an emerging pathogen disinfectant for those interested in corona. And we're more sustainable. You can find more information at nadcctablet.com. So thank you, everyone, again, for joining, and look for the series that'll be archived.
Thank you as well.
All right. Thank you, and goodbye, everyone.
Bye.